Hello, friends on the internet. We've got something special for you today in AP Bio. We're studying Mendelian Genetics. That's right. And since we're talking about how traits are passed from one generation to the next, I learned a few things this weekend I wanted to share with you. Yes, I'm not going to give this all away because I think I'm going to make a special episode on this. It's so good. But it turns out that I've got some pretty famous relatives. One famous relative, you'll have to wait for the rest of these. One of my famous relatives was Anne Alcock. She was my ninth great-grandmother. And if you look her up on the Wikipedia, she was killed in the Salem witch trials. She was accused of being a witch. You know those two girls who are going around accusing everybody of being witches? Yeah. She was one of the ladies that got accused of being a witch. They tortured her a bunch. She finally, I don't know if she admitted to it or what. They killed her and one of her daughters. Fortunately, one daughter survived. Well, I mean, maybe more than that. But one of the daughters that survived had some babies. They had some babies, and they had some babies. And eventually, I was one of the offspring. Yes, it's a beautiful thing. I also found out that my wife and I are 12th cousins. And I'm pretty sure if you're of any Anglo type of history that you're probably kin to one another. Now, I'm going to give you all the skinny on this. The website is called familysearch.org. I think our friends from the Latter-day Saints are the ones who came up with this website. It's free. You can get on there, and if you enter someone that you know that's deceased, like a grandparent or great-grandparent, and where they were born, possibly, or when they were born, around the time, there's a good chance you might find some of your ancestors too. Now, I don't know if there's a lot of non-Anglo people on this family search, but there are lots of thousands and thousands of people. So I don't know if you're like of African or Asian or Hispanic descent, if they're going to have a lot, but because I don't have any of those descendants, but or ancestors. But it's worth a shot. My wife threw in her grandmother who immigrated and ran away from home from Greece, and she was a first generation immigrant, and she happened to be on there. And my wife's like, "How's this on here? And who put this on here?" And she didn't know, and it had her old world name, not the one that she took when she got married and came here. So who knows what might be on there? And like I say, I've got a lot of special surprises that I found on there. Basically, I just went down like one rabbit hole after another. I took like my grandma and just kept going out as far as I could go. Some of these descendant lines that they've got mapped out go back to like the 11th and 12th century. Like it's like that far back. So there's like a whole lot of descendant information running around for those people who are at least of British descent, which is where a lot of my descendants was from. There was also an Indian chief in there, and so lots of fun stuff. And I may or may not have an ancestor of royalty. You'll find out more later. Yes, it's true. I knew I wasn't very humble for a reason, and now I know why. That's true. Okay, today we're starting Mendelian Genetics Part 1 in AP Bio. We're in our books on page 267 for those of you in Campbell. And Mendelian Genetics is in reference to whom? Gregor Mendel, the father of genetics. He was a monk. He didn't have any wife. He didn't have a wife. He didn't have any kids. So obviously he didn't have anything to do. And he's like, I'm going to study some stuff. And he cooked, and he's like dealing with these peas and these characteristics of peas. And it's like, I wonder how these traits are passed on in these peas. I think I know something. I see something. Let's see what happens. So he bred the peas together, and he found lots of characteristics and lots of traits and how those characters were passed from one generation to the next, okay? So that's what we're going to be talking and working through. We're going to do another practice plasmid, at least work on it, which I'm sure will be a barrel of giggles. And I think that's probably all we'll have time for today. Oh, we'll do some practice Punnett squares for those of you out there in biology land trying to figure out how to construct and make Punnett squares. All right, important concepts from previous unit. Walker, read those three.
Genes are DNA segments that are inherited from parents during reproduction. The gene is the blueprint for making a polypeptide protein. Proteins are made expressed by the processes of transcription and translation through protein synthesis.
I don't know if you and people on the internet can hear those roosters in our courtyard, but the rooster in the courtyard, Hector, he's getting after it. It looks as though we're gonna incubate some eggs at the end of this month, start incubating, and hopefully we'll have some baby chickens before spring break rolls around.
Mr. Mack, they're gonna all think that we're dicks because we have roosters and all.
Well, as in many other aspects of my life, Walker, I don't care what people think. That's okay. I bet anybody that doesn't like it is just jealous they don't have chickens at their school in their courtyard. But I bet most of you appreciate it, and y'all also wish that you had chickens laying eggs. I ate a lot of those eggs, and those jokers look like they were fertilized, so I think we're gonna be good to go. All right, genes. So segments of DNA that are passed on from parent to offspring. So things that make you unique, you got those things from your parents. Now, speaking of my wife being my 12th cousin, I looked it up. Once you go past about like second cousin, I mean, like it's relatively like the DNA that you share is almost, it's hardly anything. I think, siblings are going to share about 50% of the same DNA on average because you're getting information. You've got 4 grandparents each, but your parents are passing off information to you. And so you're not getting everything from every grandparent. Like your mom is making eggs, not the kind I made from the chickens, but she's making them, and the DNA is separating out through independent assortment to make these unique haploid cells. So you can't get all of the DNA that she had, you can only get half of it. First cousins, I think it's at around 12.5% of the same DNA. Second cousins, I think it goes down to like maybe 3% or something. And then once you get past that, it's pretty small. It's like below a percentage point. And so I think in Alabama it's legal to marry your second cousin maybe. Now, that's maybe a little uncouth, but pretty sure somebody told me in the other class after, third or later, it's not that much. You're not that close. I'm going to let you people out there on internet land make a judgment call on that one, though. Let's just stop talking. So, now, you pass on genes that are on your genome, right, from one generation to the next that affects what your offspring will look like. And we also talked a little bit about epigenetics where, that's where you're actually passing on switches where you turn on or off a gene based on methylation, where methyl groups get added to different genes, which turns them off. That can be passed on. So that can also affect your offspring and their outcome. So that's another interesting kind of factor we're not really going to get into in this section, though. All right, Gregor Mendel, we talked about him. Characters, what is that, Tay?
An inheritable physical feature. This is a characteristic such as eye color or hair color.
Okay, so characters are inheritable features, eye color, hair color, skin color. I'm going to tell you guys right up front that we're going to do some gross oversimplifications of genetics in here. For example, eye color, hair color, skin color. These are things that are controlled by either multiple genes or multiple alleles, like multiple forms of a gene. These aren't simple things like with Mendel and his pea plants, you had like tall variety or tall allele, which is a different version of the gene, or short. It's one or the other. So most of the traits he looked at was like an either or. It was this trait or it was this trait. Whereas if you're talking about hair color and eye color and skin color, there are multiple things that are genes that are going into deciding the outcome of how that's going to appear or show up. Does that make sense? So just like it's not as simple as if you have, a black person, a white person, they're going to have, a similar baby every time, like a mixed baby, right? You can have a variety of skin tones that result. I remember a few years ago they had a white woman and a black guy, and they had twins. They were fraternal twins. One of them looked very light skin, almost like white, like my skin, and then one had very dark skin. So it was very like a contrast between the two twins. You would think they would have just been kind of a mixture normally in the middle. So that's because there's a lot of genes, and it really just depends on which genes their offspring inherited. And there's actually a Punnett square in your book that kind of goes into skin color we'll look at just to show you how complicated it is. Traits. What are traits, Katie?
This is a variation of characters such as blue colored eyes or black colored hair. This requires inheriting 2 alleles, one from each parent.
Okay, so the character is an inheritable physical feature, and the trait is the variation of that. So what are some variations on those traits and how they're inherited through those alleles? The note writer says it's inheriting 2 alleles. A lot of times you're going to have 2 different forms of that gene because you get like one from one parent, one from another. I mean, you can inherit 2 different forms of it. But sometimes there's actually multiple alleles that control for a gene. So we'll get into that a little bit later and look at some different examples. Like I said, I'm going to try to keep this on the simplistic version as I can. What are alleles, Sarah? Tell us about those.
This term refers to different versions of a gene. Remember, a gene is a distinct DNA nucleotide sequence that can make one protein or enzyme. Brown, blue, green eye color. These are 3 different versions or DNA sequences of a single gene, but they all are making the eye color.
Yeah, so let's do an easy one. So with pea plants, Mendel looked at pea plant height. Let's turn over to page 270 in your book. Look at some of these traits that Mendel actually looked at here, because these are much simpler than what some of our characteristics and traits. So these are the traits that Mendel looked at. You can see here the different crosses and the ratios there. And so for like pea plant height, he's got tall and dwarf, or we just say usually tall and short. So the ratio between those two, we'll get into all that and how they're passed on and which one's dominant and which one's recessive. But basically that tall and short are just different forms of what gene? What's the gene we're looking at? The gene for, if you're looking, yeah, height. So if you're looking at the gene for plant height, pea plant height, there's 2 variations, 2 different alleles, tall and short. It could be flower color. What are the 2 different variations, 2 different alleles?
Purple and white.
Purple and white. So those are just the 2 different variations that you see. Those are the alleles. And those are the things we're going to put in the Punnett square to see how those traits get passed. Okay? B part, each trait needs 2 alleles. Dominant and recessive. I'm just going to talk about this one. What is the difference between a dominant and recessive trait?
Dominant only requires 1 allele from one parent, or recessive requires alleles from both parents.
Yeah, that's good. So dominant only requires an allele from one parent, and it is the trait that will show up. So if you have 2 different forms of that gene, 2 different alleles, it's the one that's going to show up if it's present. The only way that a dominant allele trait doesn't show up is there's 2 recessive traits, yeah. So if you inherit both recessive traits, that's the only way they're going to show up in the offspring. Good. And normally we represent this by a capital and for dominant and a lowercase letter for recessive. So when we get into Punnett squares, that's a good indication. Now, there are some traits that are neither dominant or recessive, okay? There's a couple different types of traits, and a couple different types of ways that traits are passed on, or different forms, and we'll get into that, I think, tomorrow. So that's a little more complicated. Let's go ahead and go to the next one. True breeding. What does that mean, Miranda?
These organisms only have one type of alleles for that trait.
Okay. So that means they are homozygous. What does homo mean? Same. So that means they have both those traits, whether it's 2 dominant or 2 recessive, they contain both those. So when you hear that term true breeding pairs, it just means it's good, the alleles are going to be homozygous. Okay. Hybridization. What does that mean, Mason?
This is the process of creating an organism with 2 different types of alleles for that trait, referred to as hybrids or heterozygous alleles.
Okay, hetero means
Different.
different. So you've got 2 different forms or 2 different alleles. Phenotype and genotype. So this is pretty basic here. I don't know. I'm sure you've heard of this. If you had 9th grade bio. Phenotype, think pH, physical appearance. Okay. So when you're asked to give the phenotype, that means you're being asked to give the physical appearance of whatever that trait is. Okay. you will be doing phenotypic ratios. So hopefully y'all know a little bit about ratios like 3-to-1 or 4-to-0 or 1-to-1. So we'll do that. We'll look at phenotypic and genotypic ratios once we do the Punnett squares. Okay. Genotype. What is genotype, Ashlyn?
This term refers to an organism's genetic DNA makeup for a trait.
Okay, so genotype, it's got gene in the word. It's the gene. What's in the genes? Okay. Phenotype, physical appearance, what does it look like? The hotline blinged. It could only mean one thing. No, I've forgotten what I'm doing here. Okay. If the genotype of an organism, can you always, here's a good question. Can you know the genotype from the phenotype? Mason?
No.
Why not? In what instance would you not know the genotype from looking at the phenotype? These are important questions you should ask yourself there on the internet. You might know in here?
If there's a mutation.
No, not necessarily. What's an example of when you might not know the genotype from looking at the phenotype?
If they both have like the dominant gene, or if it's like one dominant and one recessive, you wouldn't notice it. Like the dominant would still be like the phenotype.
Yeah, so if you've got the dominant gene, if you're just looking at that dominant phenotypic trait, you're not going to know what the genotype is just from looking at it. Either if it's too dominant, because you're not going to know if there's either too dominant or if there's a dominant and recessive. So when we get into making pedigrees on probably Wednesday, we'll talk about that. Sometimes you just got to leave your pedigree like this or this because you don't know which one it could be. Okay? Good job, Ashlyn. So, Punnett squares. Punnett squares. Why do we do Punnett squares? I know y'all have already had 9th grade bio in here. Does anybody remember why?
Because it just shows the possible outcomes of the genotype.
Okay, you're making a Punnett square to look at the possible probability of the offspring. What does probability mean?
What is the chance of getting them?
Probability, excuse me, is the likelihood that some event will occur. Good. So we're looking, and we'll often have either a ratio or a percentage associated with that. You can do monohybrid crosses, which is what we're going to do mostly. Dihybrid crosses are where you're crossing two traits. Y'all remember those where you have like 16 squares? Those are a real pain in the butt. We'll do 1 or 2 of those just because I know you're going to see 1 or 2 of those on the test and probably a quiz. But we won't spend too much time on there. Trihybrid crosses is like multiple traits, and I don't know why anyone would do that. You guys are going to do a thing where, an activity where you have like, here's the parent that has these 4 genes, 3 or 4. This parent has these 3 or 4 like genotypes, okay, for different genes. Here's the offspring. What's the likelihood this offspring comes from these 2 parents? And so there's actually a shorthand way of figuring that out without actually having to do a gigantic Punnett square with 64 or whatever would come next, 4 times 4. 4 to the 4th power, can't even go that high. What is 64 times 4? 200 and something. 56. All right. Law of segregation. What is segregation, Tay? No, I mean, just tell me what segregation means.
Separate.
To separate. Now, I got a funny segregation story. My daughter the other day, she was saying, talk at the table, and she's like, do you know who Ruby Bridges is? You know who that is? Who is it, Tay?
She desegregated school in Arizona and started the movement for desegregation of all schools around the United States.
Yeah, so she was like an elementary school kid, right?
She's like 15, 16.
Oh, was she that old? I didn't think she was that old. Oh, anyway. Well, my daughter's like, she was the first one to disintegrate in schools. And I was like, you mean like the first one to like in, what did she say? She said disintegrate, and that's all I got lost on. I don't know what she was trying to say desegregate, I think. And she said disintegrate. And I was like, let's hope not. If that happens, we haven't made much progress. Okay, law of segregation. So let's go back through these. And basically this law of segregation and the law of independent assortment is talking about how chromosomes and sister chromatids separate. So, what happens that's significant in anaphase 1 of meiosis? No, crossing over does not occur in anaphase 1. When does crossing over occur, Mason?
Is that interface 2?
Nope. Y'all are going to fail. When does crossing over happen? What? What'd you say?
No, I don't know.
Prophase 1. Is that what you said? Okay, so prophase 1 crossing over occurs where, remember our gang sign? So the DNA crosses over, DNA gets exchanged on the chromosomes. We're going to call ourselves the tetrads because of, the 4 different chromosomal units. That was my gang name, but like the administration didn't approve of that. Like apparently an AP Bio gang sounds too dangerous on the streets. I'm not kidding. I had this conversation. It's actually with the previous administration though. So, what was we talking about? Gang sign. Oh, anaphase 1. What happens in anaphase 1? What happens of significance in meiosis, anaphase 1 during meiosis 1?
Whole chromosomes are being it's being changed between
What separates?
haploid and haploid.
Not sister chromatids. What?
Number of chromosomes.
Homologous chromosomes separate. Y'all need to study. So you had like chromosome 1 you got from your mom, chromosome 2 you got from your dad. They crossed over, but they're still they call them homologous chromosomes because they code for essentially the same content, like hair color, eye color, skin color, whatever. They may have different forms of those genes, but they code for the same content. So those homologous chromosomes, they separate in anaphase 1. That's how you reduce that chromosomal number down to the N value, okay? Down to 23. What happens of significance in anaphase 2? Sister chromatids separate. So that's another method of where you're separating and creating genetic variation. They talk about that in the law of segregation. And during independent assortment. Basically, both those are in reference to chromosomes and the homologous chromosomes and sister chromatids separating, reducing that chromosomal number. You wind up with cells that all have different genetic content because of that. So there you go. All right, probability, we talked about that. Like if you flip a coin, you would expect a 50-50 chance of getting heads or tails, right? So that is a certain probability. Now, you may flip a coin 3 times and get heads each time. If you flip a coin enough times, though, you would expect to probably get closer to that 50-50 mark, okay? Last thing, probability. Oh, yeah, the rules of multiplication. I'm going to come back to this one. Just remind me, this is the one where you have like a parent's genotype, the offspring's or the 2 parents, and then the offspring's genotype, and like what's the probability that this offspring will have this particular genotype, even if you're talking about like multiple genes. And it's a pretty easy way to figure that out. So we'll come back to that. What I want to work on the rest of the day is doing some Punnett squares and some working on this plasmid map I've got on the board. So if you get out a blank sheet or something to write on where you can come up with some Punnett squares, we'll work on this. Okay, guys, we're going to work on Punnett squares for a little bit, and this is kind of our basics in bio, back to things that you learned in 9th grade. But I'm going to make it a little more complicated. We're going to start out here, so see if you've been paying attention. So in our first Punnett square right here, I want you to cross. Okay, so are y'all on page 270 in your book? They got the pea plants on there. I'll go ahead and give you a heads up. So pea plants, you've got 2 variations of plant height. What are they?
Tall and dwarf.
Tall and short. It says tall and dwarf, but we'll say tall and short just for easy. Okay, so I want you to cross a homozygous tall plant with a homozygous short plant, okay? And I want you to tell me what the second generation phenotypic and genotypic ratios are. Go. So I want you to cross a homozygous tall plant with a homozygous short plant. And then I want you to tell me what their second generation offspring are going to look like and what their genes are like, okay? Now, do y'all remember what the first ones you cross are called? They're called the P generation. What do they call them P? P stands for parents. Good, thank you. Yes, so that's the parent generation. Okay. So, and then their offspring we call the F1. Y'all remember that? Nobody remembers this from ninth grade? Okay, we'll have a ninth grade lesson here. F1 stands for first filial. Filial is like the Latin term for son. So this is like your first offspring, okay, from the parents. You know what you're doing yet? You know what to cross, Walker? What did I tell? Who did I tell you to cross?
Homozygous tall.
Homozygous tall. So what letters are we going to let? Now, keep this in mind out in Internet Land or in the class. You can pick whatever letters you want to use, but the easiest thing for me to do is to assign the letter of the dominant trait. So like on the parent, I would do like big T, big T. It's homozygous tall, right? It's got two big T's. And then for a short plant, what would you use, Katie?
Oh my gosh, you can use K? Because then it'd be KT, it'd be my initial.
No. Mason?
Little T's.
Two little t's. Okay, so and then the recessive trait, you'll use the lowercase version of that. Now, you don't want to use two different letters here, okay? There will be a point, Katie, when we use two different letters, but that will be when we look at a different aspect of genetics where traits can get mixed together, like a white flower and a red flower make a pink flower. We'll use different letters then. But since we know one trait is dominant over the other, we're going to use the capital letter and then lowercase letter to show the recessive. Okay, so go ahead and do that cross. And once you get this cross, you're going to cross your F1 generation and get your F2. Okay? And then the F2, we're going to do the phenotypic and genotypic ratios for each one of those. If you're not first, you're last. All right. So what goes in here, Walker?
Crosses, so basically T, big T.
Big T, little t. Good. So that's going to be all of your genotypes, big T, little t, and all the offspring here would be what?
Big T, little T.
What's their phenotype?
Tall.
They're all tall. Okay, so all the offspring would be tall, even though you crossed a tall one with a short plant. Okay? So we know that since they're all the same, we can cross two F1s, which are going to be big T, little t, big T, little t. Y'all get this far? So you're crossing the F1 generation to get the F2 generation. So this would be like find the grandkids. Okay? So when you fill these in, it looks something like this. I always write the dominant one first, just so I can keep myself organized here. So those are your offspring. Now, what I wanted you to do is, what's the phenotypic? Actually, let's first do genotypic ratio. What's our genotypic ratio here, Sarah? 3 to 1, but what are they? Is it 3 to 1?
Yeah.
The genotypic ratio?
No.
All right, when you're doing ratios, here's what you need to do. You need to write them all out first. Okay, so you got big T, big T, big T, little t, little t, little t. Okay? Those are your three genotypes. How many big T's do we have?
One.
Okay, we got one. Let me fix this here. So we got one. And then how many big T, little T's do we have? Two. And then how many little T's?
One.
Okay, so we've got a 1-to-1 ratio. Or if you wrote it out as a percentage, sometimes you see it as a ratio, sometimes you see it as a percentage. You need to know how to do both. It would be 25%, 50%, and 25%. Okay? So, make sure you know how to do it either way. People got that? All right, now what is the phenotypic ratio, Mariah? They're all tall? All the F2 generations tall? What do they have to be tall? They got to have the dominant trait, right? How many have the dominant trait, Mariah? 3. So our phenotypic ratio, the same thing. You're going to write out your different phenotypes. Tall, short. So we've got 3 tall ones, 1 short one. Or if we were writing it out as a percentage, 75 to 25. That's what we expect. Now, does it always happen like that? No. It's just like, what about having babies? Like, let's say Katie, you and Walker have a little boy. What's the chance of you having if you have 4 kids, Katie, you and Walker, say you have 4 kids, these people aren't really in my class. What's the likelihood that, how many girls and boys did you expect to have? What?
You don't know.
You don't know the likelihood?
I mean, 50-50.
Okay, it's 50-50, right? You got a 50-50 chance of having, boys and girls. Almost like flipping a coin, right? So, let's say, Katie, you have 2 boys to start off with. That next kid, the third one, what's the likelihood it's going to be a boy?
50-50.
It's still 50-50, right? So even though the overall chance before you started was 50-50, every time you have one, it's still 50-50. So it's just like flipping a coin. You flip a coin, you got a 50-50 chance of getting heads or tails. Flip it again, you may have heads like 3 times. That next time, it's still a 50-50 chance. Okay? So make sure you take note of that. All right. Plasmid mapping. We've got, for those of you on internet land, we're going to do a plasmid map here. Now, first thing I want y'all to do is fill out what the gel would look like with these digests taking place. Let me see if I can get this sorted out. All right, I'm going to erase this. So you can draw a little gel out or a big gel out probably. All right. Over here on the right side of the board, we've got some different digests. So, let's see. Is this going to show up? Yeah. All right. So you're going to have 6 wells. 1, 2, 3, 4, 5, 6. So that's where we're putting our DNA in. Okay. And the DNA, the plasma that's being digested, how big is it? You might tell by looking at that. 87.2. So when it's cut just one time, it's 87.2. So that's how we know how big the plasmid is. So when you put the DNA in here and you cut it with BAM high, I'm just going to say BAM, it's going to go down and it's only going to be, it's going to be 87.2. Probably not going to go that far, right? Pretty big. Same thing when you just cut it with ECO. Same thing when you cut it with, what's the other one? Hind. It's only going to cut it into 1 strand, and they're going to be 87.2 kilobase pairs long. Okay? So, the next thing, we're going to cut it with 2 different restriction enzymes. When you cut it with the ECO, I'm just going to put the first letter here so I can see. ECO and Hind. It's going to break it up into a 79.6 segment and a 7.6 segment. Okay? So this is what it would look like if you put this plasmid, mix these restriction enzymes, and then put it in this gel and it's separated out. So these are the different segments that would result. Okay? Now, the next one. If we cut it with ECO R1 and BAM, the restriction enzymes here, it would cut it where there's a 72.2 segment and a 15 segment. And then the last one, the BAM and the Hind, it's going to cut it into a 64.6 and a 22.6. Okay? So these restriction enzymes are cutting this DNA at different points. Okay? Winding up with multiple segments there. All right, now, how do we create a plasmid map from this? I'm going to give you the simple answer. It's going to be trial and error. Okay? So most plasmid maps are, you're just going to figure them out trial and error. So we got a circle here is our plasmid. We know it's 87.2 kilobase pairs. Okay? And we know that it gets cut in 3 different locations because we're using these 3 different restriction enzymes. So that should be pretty easy to figure out, right? Only 3 places, 3 restriction enzymes. How hard is that? Well, the problem is you got to figure out where it cuts them because they got to be in a certain order. Okay? So for instance, we can just start with the first one. Let's say BAM cuts it right here. And then you've got an 87.2 segment. Okay? But now you've got to figure out where does ECO cut it? Where does the hind cut it? These are things that you have to figure out. Does it cut it? Does their ECO go over here? Does it go over here? Does hind go in between it? Does it go on the outside of it? This is what you have to figure out. So I would use a pencil and you'll kind of trial and error this. The easiest way to do this is to try out like where maybe ECO and hind is. And then you need to look at these numbers right here. So if you were to cut it, like once you get like a couple practice cuts in there, then look, is it going to cut it, if this is correct, is it going to cut it into these fragments? If you look at them just with the two individual cuts. Okay? So, I know that's confusing, but I'm gonna let y'all practice, then I'm gonna run y'all through it. So, we're practicing trial and error here. So Katie, she cut it with BAM up here, and so she's gonna do ECO as her next cut right here. Okay. So if you cut it with these two right here, what has to be the number in between here? 15. And then all of this right here would be 72.2, right? Okay, 72.2. Okay, now you've got to figure out hind. Where are you going to cut it? It's either got to be cut on this side, in the middle, or somewhere over here.
I put it on the right side of BAM.
You put it on the right side of BAM?
Possible to be 7 from.
So if you put it, yeah, so listen to what he's saying. If you put, let's see, we can do it here. Let's say you cut with the hind over here, and you put it, so when hind and BAM, you've got to wind up with a fragment that's 22 and 64.6. So that would mean this would have to be 22.6, and this would have to be, from here all the way around would have to be 64.6.
It'll have to be super close to ECO since there's a 7.6 here.
Okay, so yeah, so when you cut with ECO and hind, you wind up with a 7.6 fragment and a 79.6 fragment. So where do you think it would go?
The left of E.
The left of E. So if we do it with the left of E, you can have your 7.6 fragment right here. And then when you add all this up all the way around, what do you wind up with? You might know?
79.6.
79.6. And so if you figure it up, this part right here is how big? Does anybody know?
64.6.
64.6. So as you go through and check them, like let's say we do it with BAM and hind here. If you just look at BAM and you just look at hind, you have a segment that is 22.6, 64.6. So that checks out. If you look at ECO and BAM, you wind up with 15 and you wind up with 72.2. If you look at ECO and hind, just ECO and hind, you wind up with a 7.6, and if you add these up, you've got 79.6. So all of them check out.
That makes sense.
That makes sense? So it's not easy. The not easy part is you don't know. You've got to plug some stuff in and you got to check it. And if it doesn't work, then you got to figure out what went wrong. So it is a little bit trial and error. Okay?